Usually when we talk about a medical diagnosis, there's this expectation of, like, mechanical precision.
Right, like it's a simple math problem.
Exactly. You break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is.
Because when you're dealing with physical trauma like a fractured bone, the physical reality perfectly matches the visual evidence. I mean, it's binary, broken or not broken.
But then you step into the world of complex diseases, like cancer, and suddenly that x-ray machine is useless. We are looking at a diagnostic landscape that is, well, the absolute definition of muddy waters.
Highly chaotic.
Totally. So today we're clearing that mud. Welcome to the deep dive. We are unpacking a 2025 AI framework called SIDISH. It's detailed in a fascinating paper from Nature Communications.
It really is a game changer.
And our mission here for you listening is to understand how scientists are finally bridging the massive gap between microscopic cellular details and big picture patient survival data, all to find cancer's deadliest cells.
Yeah, and solving this requires overcoming a massive scaling problem in biology, one that has frustrated researchers for over a decade.
Okay, let's unpack this scaling problem, because we have two very distinct ways of looking at genomic data, right?
Right, and historically, they simply refuse to talk to each other.
So for anyone who has tried to keep up with genetics, you've probably heard of RNA sequencing. And I want to frame this for you as a choice between a smoothie and a fruit bowl.
Oh, I like that.
So for years, large-scale studies have relied on bulk RNA sequencing. That is your smoothie. You take thousands of cells from a biopsy, blend them all up, and analyze the total genetic expression.
Right, so you get the overall nutritional profile, but
if there was one single, highly mutated, toxic strawberry in that blender, you'd never see it.
Yeah.
It is completely masked by the blend.
Exactly. The alternative to that is single-cell RNA sequencing, or cRNA-seq, and that is your fruit bowl.
Where you see everything.
Yeah, instead of blending the tissue, this technology isolates every individual strawberry, blueberry, banana. It reveals the cellular heterogeneity, basically the unique, widely different personalities of every single cell in a tumor.
Which sounds like the perfect solution, until you look at the price tag.
Yeah, buying a high-definition fruit bowl for millions of people isn't just difficult, it is financially impossible.
Just to put some concrete numbers to this for you, as of 2025, running single-cell RNA sequencing costs roughly $5,000.
Right, and that's just to analyze 20,000 cells from one single sample.
$5,000 for one sample.
You just cannot scale a $5,000 per sample test across large clinical trials that involve thousands of patients.
It's just not going to happen.
No. And as a direct consequence, single-cell studies are typically very small. And because they're small, they lack the one crucial piece of clinical metadata that actually matters.
Long-term patient survival outcomes.
Exactly.
So we are stuck in a paradox. We have the cheap, scalable smoothie data, which is tied to massive databases of real-world patient survival. We actually know who lived and who died. And then we have the expensive fruit bowl data, which shows us the precise cellular mutations, but without enough patients to know if those mutations actually killed anyone.
Right.
So why has it been so incredibly hard to just merge these two data sets computationally? I mean, we have powerful computers.
Well, the scientific community has certainly built tools to try. Like previous algorithms, Scissor and Scab are good examples. They attempted to map the high-resolution single cells onto the bulk survival data.
But they use something called Pearson correlation, right?
Yes, they did.
And my memory of statistics is a little rusty, but Pearson correlation is basically looking for a straight line. Like if this gene's activity goes up, the patient's survival time goes down in a perfectly linear fashion.
And that right there is the fundamental flaw. Gene regulation is almost never a straight line.
It's messy.
It is a wildly complex, nonlinear biological network. I mean, gene A might turn on gene B, which then suppresses gene C.
But only until a certain stress threshold is reached.
Exactly, at which point the whole cycle reverses. Assuming a linear relationship completely flattens the intricate, chaotic dynamics of how cancer cells actually behave.
Okay, but I read that another tool, called DGAS, tried to fix that linear problem by using a neural network. But the paper we're looking at today suggests DGAS created a totally different mess.
Yeah, so DGAS was capable of capturing nonlinear relationships, which was good, but its architecture forced both the single-cell data and the bulk survival data into a single, shared latent space.
Meaning what, exactly?
It essentially tried to compress two wildly different languages into one generic alphabet.
Oh, I see.
And when you do that, the strong, vital clinical survival signal gets entirely diluted by the sheer noise of the cellular data.
It gets washed out.
Exactly. But honestly, the most glaring limitation of all these older tools is that they use what's called a single-pass integration strategy.
Meaning they look at the data once, make a guess, and call it a day.
Yep, one and done.
Which finally brings us to the breakthrough. The framework is called SIDISH. That stands for Semi-Supervised Iterative Deep Learning for Identifying Single-Cell High-Risk Populations.
Quite the acronym.
Right. So if the old tools mashed everything together in a single pass, how is SIDISH actually learning the difference between a harmless cell and a lethal one?
Well, the magic word in that acronym is iterative. SIDISH doesn't just make one pass. It utilizes an iterative feedback loop.
A loop.
Yeah, it's constantly translating back and forth between the high-resolution single-cell data and the bulk survival data.
Okay, so it looks at the single cells, extracts their genetic patterns, and tests them against the survival outcomes.
Right. And then it uses the results of that test to go back to the cellular data and say, okay, these specific genes seem to correlate with death. Let's weigh them heavier and test again.
Oh, think of it like going to the optometrist. You look through that massive machine and the doctor flips a lens. Better one or better two.
That is a perfect analogy.
You give feedback. And based on that, they flip another lens, refining the prescription over and over until the blurry letters become crystal clear.
And SIDISH is doing that with millions of data points, flooding mathematical lenses until the deadliest cancer cells snap into perfect focus.
Wow. Okay, we are right around the 5-minute mark of this deep dive, so let's do a quick mini recap for you listening, just to make sure we've got the foundation locked in.
Sounds good.
So, bullet one single-cell RNA sequencing gives us micro detail, but bulk RNA gives us clinical scale.
Right. And Bullet 2 older tools tried to combine these, but they either forced linear relationships or diluted the data entirely.
Which leads to Bullet 3. SIDISH solves this by using an iterative neural network, allowing it to keep both the detail and the clinical relevance.
Spot on.
So here's a question for you listening. If you were designing a system to find the needle in the haystack, would you rather look once or have a system that learns what the needle looks like a little better with every single pass?
I think the answer is pretty clear there.
Totally. So now that we know what SIDISH does, let's lift the hood and look at exactly how it works, mechanism by mechanism.
Right, because to really appreciate how it achieves that focus, we need to look at the underlying architecture.
Yeah.
The framework operates in 4 distinct phases.
Okay, let's trace the signal through the machine. Phase 1 is called extraction, and it involves something called a variational autoencoder, or VAE.
Yes.
But the part I need help translating is this. The paper says the VAE uses a zero-inflated negative binomial reconstruction. What on earth is a zero-inflated negative binomial, and why is it so important here?
I know, it sounds incredibly dense, but it actually solves a very practical physical problem with sequencing machines.
Okay, like what?
Well, when you are analyzing single cells, the machines often fail to detect a molecule of RNA, even though it's physically there in the cell.
Oh, so it just misses it.
Exactly. This creates a false zero in the data, what geneticists call a dropout.
Like listening to a radio station where the signal keeps cutting out for a millisecond. The music is still playing at the station, but your receiver recorded silence.
Perfectly stated. And if an AI takes those zeros literally, it thinks the gene is turned off, which corrupts the entire analysis.
Because it's working with bad data.
Right. So the zero-inflated negative binomial is a specific statistical model that teaches the neural network how to distinguish between a true zero, meaning the gene is actually off, and a dropout zero, where the machine just missed it.
Oh, wow. So by correcting for those dropouts, the VAE can compress this massive, noisy genetic data into a clean, mathematically manageable latent space.
Exactly.
So the VAE acts like a biological ZIP file. It compresses millions of complex genetic interactions down to their core, essential patterns without losing the signal to background static.
That's a great way to put it. And once we have that clean ZIP file, we move to phase 2, which is survival prediction via transfer learning.
Okay, this is where we bridge the gap.
Right. The system takes the encoder weights the core patterns learned in that biological ZIP file and transfers them directly into a deep Cox regression model.
Now, I've seen Cox models used in a lot of clinical studies. They basically measure the risk of an event happening over a specific timeline, right?
Yeah, that is their exact purpose in medical statistics. A Cox model predicts time to event, which in this context is patient death.
Heavy stuff.
Yeah. The model is trained on the cheap, scalable bulk RNA sequencing data and the actual patient survival outcomes.
So by transferring the high-resolution cellular pattern into this survival model, SIDISH is effectively teaching the macro-level clinical data how to see micro-level details.
Exactly. It's linking the two worlds.
Which sets up phase 3 risk prediction and stratification. The Cox model is now assigning a continuous risk score to every single cell in every patient.
Yes, but you can't just hand a doctor a spreadsheet of continuous numbers.
Right, you need to draw a hard line in the sand and say everything above this line is a lethal threat.
Exactly. And the paper says SIDISH draws this line using a Weibull distribution.
Okay, wait. Why a Weibull distribution? Why not just use a standard bell curve?
Well, because a standard bell curve assumes that data is distributed symmetrically around an average. And that just doesn't reflect how biological failure works.
How so?
A Weibull distribution actually comes from the field of reliability engineering. Engineers use it to model time to failure.
Like predicting exactly when a car engine belt will snap after enduring cumulative stress.
Precisely.
That is incredibly morbid, but it makes perfect sense. The biological system is the engine, and the cancer is the cumulative stress.
Right. It models a reality that risk isn't symmetric over time, it compounds. By fitting the risk scores to this time to failure curve, SIDISH can set a statistically rigorous threshold.
It separates the highly dangerous, high-risk cells from the benign background cells with pure mathematical precision.
Exactly. Which brings us to the final and arguably most important part of the machine, phase 4.
The iterative weight updates.
Yep. This is the optometrist of flipping the lenses. The system uses SHAP values, which stands for Shapley Additive Explanations.
From my understanding of machine learning, SHAP values are essentially a way to force a black box AI to explain its homework.
That's exactly what they do. They assign a specific contribution score to every single feature the AI considered.
So in this case, SHAP values quantify exactly which individual genes contributed the most to a cell being pushed over that Weibull threshold into the high-risk category.
Right. So once SIDISH identifies the most dangerous cells, the SJT values highlight the specific genes driving that danger.
And then this is the iterative part. It feeds those specific gene highlights back into the phase 1 ZIP file.
Yes, updating the weights so the model pays even closer attention to them on the next pass. And it just keeps looping.
It loops until the genetic features perfectly align with the survival outcomes. It doesn't just find a loose correlation, it actively hones in on the exact molecular drivers of death.
It's a truly elegant system.
So think of it like this. Phase 1 is the detective finding clues. Phase 2 is handing those clues to a judge, the Cox model, who looks at the outcome, survival. And phase 4 is the judge sending the detective back to look for more specific evidence.
That is a perfect visualization of the 4 phases.
Awesome. Okay, we've hit another 5-minute mark. Time for a MIDI recap. Bullet 1, phase 1 compresses the data using a VAE biological ZIP file. Bullet 2, phase 2 connects it to survival data using a deep Cox model.
And bullet 3, phase 3, defines the danger threshold using a Weibull time to failure distribution.
Which leaves bullet 4, SHAP values, create an iterative feedback loop to refine the focus. So my question for you listening is this: which of these 4 phases do you think acts as the true brain of the operation?
Oh, that's a tough one to answer.
It really is. So if you're listening to this and wondering, okay, the math sounds brilliant on paper, but does it actually work in a physical body?
Yeah, theory is one thing.
Right. That is exactly where the researchers went next. They unleashed SIDISH on real-world cancer data sets. Let's look at the clinical case studies, starting with pancreatic ductal adenocarcinoma, or PDA.
And pancreatic cancer is notoriously aggressive. Survival rates are historically dismal.
It's terrifying. So what happened when they ran the data?
Well, the researchers fed SIDISH a massive single-cell data set of pancreatic tumors. The algorithm sifted through the noise and identified 3,623 high-risk cells.
Which represented about 8.6% of the total cell population.
Right, but the crucial finding wasn't just the number, it was the identity. Over 55% of those high-risk cells were classified as type 2 ductal cells.
And what makes a type 2 ductal cell so dangerous in this context?
In pancreatic cancer, type 2 ductal cells are known drivers of aggressive tumor progression. They heavily influence how the tumor interacts with its environment.
But the real power of SIDISH emerged when they mapped these cells back to specific individual patients, right?
Yes. The paper contrasts two distinct cases, patient T20 and patient T3.
I have the numbers here. So in patient T20's tumor, SIDISH flagged 31.5% of their cells as high risk.
Almost a third of the tumor was actively lethal tissue.
Contrast that with patient T3, where only 1.5% of the cells were high risk.
But it goes deeper than percentages. The algorithm flagged massively elevated expression of specific genes in patient T20, particularly one called KRT17.
KRT17, what does that do?
It is a fascinating and terrifying gene. It doesn't just sit there, it actively alters the immune topography of the tumor.
Meaning what?
It creates an immunosuppressive environment. It's essentially building a chemical wall that prevents the patient's immune system from attacking the cancer.
Wow. So it's cloaking the tumor.
Exactly. And when the researchers took patient T20's specific genetic signature and cross-referenced it against the massive TCGA bulk database,
which holds thousands of real-world survival records.
Right. The data confirmed the AI's grim prediction. Patients with that T20 signature had vastly worse survival outcomes.
So the algorithm successfully translated microscopic single-cell anomalies into a macro-level clinical prognosis.
It did. And they didn't stop at pancreatic cancer. They applied it to lung adenocarcinoma, or LUAD, but with a twist.
Oh, right. They fed the algorithm a dataset completely stripped of human-provided cell labels.
Yes. They basically blindfolded the AI, dumped raw, unlabeled genomic data into it, and said, find the danger.
Because they wanted to prove the framework didn't require human bias or hand-holding?
Exactly. And even without labels, SIDISH independently pinpointed 168 high-risk cells. It determined that the danger in these lung cells was being driven by genes like ENO1 and LDHA.
Okay, let's drill into the biology of that. Why did the AI zero in on ENO1 and LDHA? What are those genes doing inside a lung tumor?
They are the primary engines of a process called anaerobic glycolysis.
Which means burning energy without oxygen, right?
Right. As a lung tumor grows aggressively, it often outstrips its own blood supply. This creates regions of hypoxia areas with almost zero oxygen.
And most healthy cells would simply die in that environment.
But ENO1 and LDHA allow the cancer cells to reprogram their metabolism, so they survive and continue dividing without oxygen.
So the cancer is essentially learning to hold its breath while it expands.
Yes. And SIDISH discovered the survival mechanism entirely on its own just by looking at the math.
That is wild. They also looked at breast cancer, specifically the TNBC subtype, right?
Yes, and it found high-risk cells mainly in cancer epithelial cells. But what it really highlighted there was massive interpatient variability.
Like one patient having 15.5% high-risk cells, while another had only 2.3%.
Exactly. It shows how personalized this disease really is.
But if I'm a skeptical researcher, my immediate concern here is overfitting.
Naturally.
How do we know SIDISH isn't just memorizing the weird, highly specific biological quirks of patient T20? How do we know it's discovering a universal law of cancer biology rather than just hyperfixating on noise in the training data?
Overfitting is the bane of all machine learning, which is why ablation studies and independent validations are critical.
So how did they prove it?
But the researchers took the exact marker genes that SIDISH flagged as dangerous in the initial training data, and they applied them to completely isolated, independent patient cohorts.
Oh, wow.
For example, they tested the pancreatic markers on the GSE 224564 cohort, and they tested the breast cancer markers on the CALDAS 2007 cohort.
Which are entirely different groups of patients sequenced in different labs, likely years apart.
Yes. And in every single independent cohort, those same marker genes successfully stratified the patients.
So the high-risk signature wasn't a fluke?
No, it proved to be a universal phenotype for poor survival. The algorithm found the fundamental architectural flaws driving the disease.
Incredible. Okay, time for our final mini recap.
Go for it.
Bullet 1 in pancreatic cancer, SIDISH pinpointed type 2 ductal cells and accurately stratified patients like T20 versus T3. Bullet 2 in lung cancer, it found danger cells driven by hypoxia, even without human-provided labels.
And Bullet 3, it proved its markers generalize across totally independent patient cohorts, avoiding the overfitting trap.
So a question for you to ponder. If you were an oncologist treating patient T20, how would seeing that 31% of their cells are high risk change your urgency compared to treating patient T3?
I imagine it would completely change your treatment plan.
Absolutely. So we have an AI that can reliably find the needle in the haystack and prove that needle is dangerous. But a tumor isn't a spreadsheet. It's physical, three-dimensional tissue inside a human body.
Right. Identifying the lethal cells on a screen is one thing, but we need to know where they're actually hiding in the patient.
And that requires mapping the digital data back onto physical reality, which is the focus of spatial transcriptomics.
Because cells don't exist in a vacuum. They exist in complex neighborhoods called the tumor microenvironment.
So how do they map it?
The researchers utilized a cutting-edge spatial technology called 10x Xenium on pancreatic tissue.
Oh, so it essentially takes the biological ZIP file and uses it to draw a physical map of the battlefield.
Exactly. And the spatial mapping revealed that 67.5% of the cells SIDISH flagged as high risk were physically located directly inside the regions of malignant tumor cells.
They weren't just scattered randomly.
No, they were clustered deep within the most dangerous active zones of the tumor.
Which perfectly sets the stage for what I consider the absolute climax of this research. It's an internal module the researchers call Silico Perturbation.
Oh, this part is brilliant.
I look at this as a digital flight simulator for cancer drugs.
That's the best way to describe it. In biological research, a perturbation experiment means you physically knock out or alter a gene in a lab dish to see if the cell dies or changes behavior.
And in doing this physically requires immense resources, months of time, and millions of dollars.
But SIDISH bypasses the physical lab entirely. It systematically simulates knocking out specific genes inside its own neural network.
To see if the mathematical profile of a high-risk cell reverts back to a safe background state.
Exactly. It's testing biological therapies inside the matrix.
And the results they got when they ran this flight simulator on the lung cancer data are staggering. The algorithm simulated thousands of gene knockouts, and it output a score showing which ones neutralized the danger cells most effectively.
Right, and the two highest scoring targets were the genes VDFA and MAP2K1.
Now, are those actually druggable targets in the real world, or is this just theoretical computer output?
This is where the computational theory crashes into clinical reality. VDFA and MAP2K1 are not just theoretical targets.
Okay.
They are the exact biological mechanisms targeted by bevacizumab and trametinib, two highly successful, widely used, FDA-approved cancer therapies.
Wait, really? An algorithm fed nothing but single-cell spreadsheets and bulk survival timelines, independently rediscovered multi-billion-dollar pharmaceutical strategies.
It did. It looked at the complex, chaotic web of lung cancer biology, did the math, and essentially said, if you want to stop this, you need trametinib.
Magnitude of that cannot be overstated.
It validates the entire framework. If the AI can accurately rediscover existing drugs without prior knowledge, we can trust its predictions for novel targets that we haven't discovered yet.
And it goes further than single targets, right? It can test combinatorial power.
Yes. They used the simulator to digitally knock out two genes simultaneously, MAP2K1, and another gene called FN1.
And because it's digital, it literally takes seconds instead of years.
The model demonstrated a massive synergistic effect. Knocking out both genes simultaneously reduced the high-risk cell population far more effectively than targeting either gene alone.
Think about the applications of that for a moment. Pharmaceutical companies spend billions of dollars running physical clinical trials, trying to figure out which drug combinations won't just fail or cause extreme toxicity.
It's a huge bottleneck.
If you can digitally simulate thousands of combinatorial therapies on a specific patient's tumor profile before they ever swallow a pill or sit in a chemo chair, you are fundamentally altering the timeline of precision medicine.
You really are. But as with all bleeding-edge computational frameworks, we do have to acknowledge the current limitations.
Right. It's not magic.
No. SIDISH is exceptionally powerful, but it is also exceptionally hungry.
Hungry in terms of computing power.
Yes. The iterative deep learning process constantly looping between the VAE and the Deep Cox model, it requires immense graphical processing unit resources.
You're not running this on a hospital standard desktop computer.
Definitely not. Furthermore, the framework currently relies exclusively on RNA transcriptomic data, and biology is multi-layered.
Right. RNA is just the messenger. It doesn't capture everything.
Exactly. A truly comprehensive model would need to integrate multi-omics, meaning proteomics, which looks at the actual proteins executing the functions, and ATAC-seq, which measures the physical accessibility of the DNA chromatin.
So incorporating those layers is the necessary next frontier for this technology.
It is.
Well, even with those limitations, The leap forward is undeniable. If you think back to that broken X-ray machine we talked about at the start, the murky, chaotic waters of complex diseases, tools like SIDISH, are finally providing a lens that can cut through the mud.
It doesn't just categorize cells. It anchors those microscopic anomalies to the harsh reality of patient survival. And then it provides a digital sandbox to figure out exactly how to destroy them.
We are transitioning from an era of biological observation into an era of deterministic computational intervention.
A new era of medicine.
And as we wrap up, I want to leave you with a thought that builds on that transition. If an AI framework can digitally simulate the exact genetic knockouts required to neutralize a specific patient's high-risk tumor cells, entirely in silico, how long will it be until the phrase trial and error is completely erased from the medical dictionary?
That is the ultimate goal.
Will the oncologists of tomorrow look back at our current methods of prescribing chemotherapy the same way we look back at bloodletting? Keep questioning, keep exploring the data, and we will see you in the next deep dive.